This is my disclosure. Genome-wide association studies have been employed to identify the genetic variants associated with different complex traits, and it has been applied to psoriasis for many studies. So this is a really most recent work that has revealed over 100 disease-associated loci using over 36,000 psoriatic cases. So this really gives us a comprehensive genetic architecture of the disease to help us to understand a lot of different things. But let's step back just to reveal what are the main objectives for these genetic studies. So this is a review paper like 16 years ago, but I think it's still really useful for us to just to recap what are the main goals. First, the genetic study is trying to identify what the pathology of the disease, right? Comparing case control difference, aiming to identify biomarkers, identify potential therapeutic targets. On the second goal, instead of looking at case control difference, we are more talking about the heterogeneity among cases. So this will be more subtle changes when compared with just case control difference. The goal for that would be like trying to aiming for personalized medicine, identify individuals, identify cases that could be more prone to certain comorbidities or like different rates of drug response. But we are facing different challenges. For the first aim, now we know that most of these disease variants will be non-coding. So you want to understand what are potential cell types that get involved, what are the gene targets they're going after, and what's the mechanism, right? So using only genetic data alone is not enough. On the second goal here, as I mentioned, like really modest effect size. So you could imagine the number of individuals that get into the second study would be much less than the case control study on the left, right? So again, so that's why how could we utilize multi-omics information to leverage this information to enhance the interpretation of the genetic data is like really key. So in my today's talk, I'm going to talk about some of the interpretation of the GWAS results, aiming to use some statistical genomics techniques to reveal how some of these loci that are associated with psoriasis actually play roles, play regulatory roles in keratinocytes. And by utilizing spatial omics data, trying to identify what are potential cell type origins of the cytokines that are influencing those gene targets from the GWAS signals. And then in the last part of my talk, I'm going to talk a little bit about like how we could use the genetic data to provide some translational inference. So back in the earlier phase of GWAS, we already appreciate that most of those GWAS loci are overlapping with the regulatory elements. And at that time, by utilizing different ENCODE data that using H3K27 acylation marks as an active enhancer surrogate, that are showing the psoriasis loci here are enriched among the different regulatory elements in T cells. And we also replicate that in this study, showing how like, CD4, CD8 active enhancers actually are overlapping with the GWAS loci. But what about other cell types? So to address this, okay, we utilize the most recent GWAS. So assuming this is like the most comprehensive collection up to date. And then we look into the potential gene targets in the GTEx data, so which have like a collection of genotype individuals with expression being profiled in different tissues, including skin, including blood. And then once we identify some potential gene targets for this over 100 loci, we integrate with our single cell data to try to see what are the expression profiles be like, okay, for those genes, okay? And you can see here for the heat map, for the heat map on the right here, the y-axis representing all the potential GWAS targets, and then the x-axis representing different cell types. The reddish color representing those having high expression profiles. You can see besides only the T cell dendritic cells, many of those GWAS hits, okay, they also have good expression in the keratinocytes. So our research mainly focusing on looking into the regulatory roles for GWAS signals in keratinocytes. So we implement this genome-wide, transcriptome-wide association studies utilizing our in-house data sets. So what we've done is we collect 50 individuals keratinocytes using RNA-seq to profile them. We genotype every individual, okay? And then for each of those keratinocytes, we stimulate that with different cytokines, okay? We have like 8 different conditions here. So the goal for us is actually to learn the expression patterns, okay, as a function of genetic variants in the local regions, okay? So we learned this, we model this called the genetic regulatory component of the gene expression, GREX, for each cytokines. So you can imagine that for each cytokine, for each gene, you try to model how would each of these genetic variants would try to associate that with it. And then once you learn this GREX model for each cytokines, you could then impute this GREX component in the cases and in the controls in your GWAS. Then you can actually compare that. So you're comparing this genetic regulatory component in the keratinocytes, comparing that in cases and controls, to nominate what's the potential gene targets. So what's the result? So instead of having, so this is the Manhattan plot. So instead of having genetic variants as a dot, we now actually have a gene as a dot. So remember we talked about like from the genetic variants, it's just really ambiguous. You cannot really call what are the potential gene targets would be. Now with this modeling, you're able to nominate potential gene candidates, and those are the ones that have GWAS variants play a role in keratinocytes. And then we actually apply this TWAS techniques to study multiple different complex skin conditions, including psoriasis. Here, the figure on the bottom left here representing actually different independent components, because we talked about we have 7 or 8 different cytokine conditions, right? So we look into, we just simply ask, okay, tell me among those TWAS targets and genes that we identify, how many independent inflammatory components in keratinocytes that play a role by these complex traits. So yeah, we identify like 4 to 5 different independent components. This would be like Th2 base, mostly for AD, but then we have like IL-17, TNF, mainly for psoriasis, and we have others. And then the table on the right here representing when we apply it, like how many TWAS target genes that we got from all those psoriasis loci. So indeed, we identify over 20% of these GWAS targets, of these GWAS loci that we just reviewed in the big GWAS paper, have roles played in keratinocytes. Okay, so we now know there's like TWAS target genes that have expression induced by cytokines, especially IL-17, TNF, not really surprising. But where do cytokines come from? So this is the next question that we want to ask. Okay, first, we have a list of TWAS target genes from this TWAS analysis, which we use keratinocytes to learn the model training, to do the model training. So with TWAS target genes, we just do it like sanity check. So what we're doing here is that the x-axis here representing all these TWAS target genes, and then the y-axis here representing the single cell data that we just share. So you can see that there's like majority of those TWAS target genes still have a high expression, okay, in keratinocytes. Okay, this is what we expect. I think that's just a check using independent data, so that's good. So how would the potential cell-cell interactions could potentially play roles, okay, in modulating these TWAS target gene expressions? So I'm going to switch gear a little bit, talking about the genetic application on the translational, making translational inference. So as I mentioned, there are like different ways that you can think about how to use genetic data to provide some translational applications. For example, assessing the risk of psoriatic arthritis among psoriatic individuals, asking about what's the risk of other comorbidities like type 2 diabetes, or looking at the drug response. Okay, So for each of these items that I'm listing here, we're still talking about within individuals that have the psoriasis. What are the heterogeneity, what are the variance between them? And usually this is more modest when compared to case control, okay? We are always dealing with high dimension data. So just to give you an example, this is a pharmacogenetic study that we just published like last year in BJD. We aim to look into the association between a genetic variant, between genetic variants with drug response, okay? In here is like the anti-TNF response. We're using exome chip data with the GWAS content to do these associations, like conduct pharmacogenetic study. We identified this good candidate, and then this marker itself is actually a EQTL for KLK7. KLK7 is like play roles in the proteolysis, okay? So we were saying that, okay, so now we have this connection with the gene. Can we look into the gene expression profile? So we look into the independent expression data, and indeed showing how KLK7 is upregulated in the lesional skin of psoriasis, and then upon the anti-TNF treatment, it's actually go down for expression. So then we were saying, well, maybe we want to hypothesize that there was some role played by KLK7 in modulating the TNF stimulation in keratinocytes. So we did the KLK7 lockdown and then tried to stimulate the keratinocytes with the TNF and see how some of those like pro-inflammatory response genes that are responding to the TNFs actually go down once the KLK7 lockdown. So this is actually combining the pharmacogenetic studies more upstream to the functional genomics more downstream, okay? But what I want to share with you is actually more combining things in more upstream. So as I mentioned, like that, different things that you could do. The study that I just mentioned about the pharmacogenetic studies right in here, just saying using genetic data to associate the genetic variants with the drug response. We also had a different studies in here that using RNA-seq data, okay, using the baseline expression of the RNA-seq data expression profiles to assess the risk of future drug response in the anti-TNF drug, okay, using only expression data alone. Then the question is, okay, now I was talking about all this like model effect. How could we combine this? How could we integrate some of this information? This would be really useful for the field because there are like different studies from different cohorts. If there's a way to integrate leveraging information to enhance some like drug response assessment, I think that would be really important. So it turns out that as long as we have some partially overlapping features between these cohorts, we actually could borrow information. So what we did was actually doing a variant calling from the RNA-seq data, okay? And then we have some markers that are overlapping with the genetic cohorts from the pharmacogenetic study, okay? And then we could actually, when we try to do the model training, when doing the drug response assessment, we can actually take advantage of things in here to do transfer learning to try to boost up the estimation of the parameters right in here. And what's the results? So in here, we're showing like how the different comparisons. So first, if we're using only the pharmacogenetic study, okay, the AUROC is like 0.65. Okay, so that's actually a lot contributed to the really large dimension data. And if we only using expression data from the RNA-seq or only using genetic data from variant calling from RNA-seq, we got like 0.6, 0.7, 8. But then when we use like combining data, taking information from both, okay, leveraging information to transfer learning, we got actually up to like 0.89. Okay, this is all under cross-validation. So actually, it shows that even independent studies, we may be able to leverage all this information together to enhance the predictions. All right, so with that, I'll just the conclusion. So we show that now we have a not yet finished still, but more comprehensive genetic architecture for psoriasis genetics now. Now I think it's time to really look into many of those different things that could not be answered previously. including mechanisms. But using genetic data alone is not enough. So leveraging multiple information is really key to interpret the mechanism and also look into potential targets to obtain the two goals that I just mentioned about earlier in the slides. Okay? And then we do show that there's indeed a reasonable set of GWAS loci that do play roles in keratinocytes. And we show that by integrating with spatial data. So for those GWAS loci that play roles in keratinocytes, they seem to be modulating by IL-17 TNF, and those seem to be coming from Langerhans cells, C16, positive dendritic cells, and also TC17 cells. And by integrating GWAS signals with drug response data, including the RNA-seq data that I mentioned, this actually could facilitate the pharmacogenomic study. So with that, I'd like to thank a lot of people. There's like multiple different studies in here, so especially Johan and the modeling lab from UCLA providing the pharmacogenomics data and also the spatial data. And so from my lab, Hai Han has been doing a lot of really great work for modeling these TWAS studies, and Timonga is doing the data integration stuff for the pharmacogenomic study. Thank you.